Hello! And we're talking about biological molecules. Now, why is it important to learn about biological molecules? It's important because they're what you're made of. They're what you're made of. They're what you're built out of. Now, you may remember the way we describe the levels of organization of an organism as octus: organelles, cells, tissues, organs, and organ systems. But of course, we can go lower than that. We can think about, well, we're all made out of atoms. Those atoms come together to make molecules, and those biological molecules come together to make the organelles, the small parts of a cell. Then organelles making cells, cells making tissues, tissues making organs, organs making organ systems. So at the heart of it all are biological molecules, and it's really important we know about them, but not only because of that, but also because they are at the heart of biology. They are the foundation of our understanding of what really goes on in biological organisms. Let's start off thinking about carbohydrates. Okay, all carbohydrates contain the elements carbon, hydrogen, and oxygen. You can see it in the name. Carbohydrate, carbo, carbon, hydrate, well, water, therefore hydrogen and oxygen. In fact, all biological molecules contain carbon, hydrogen, and oxygen. So carbohydrates are no exception there. And there are two types we're going to think about. We're going to think about simple sugars and complex carbohydrates. What do those terms mean? Well, simple sugars, they are soluble in water. Something is going crazy here. There we go. What a crazy animation. I don't think that was deliberate. They are soluble in water. They taste sweet, and they might be made of one unit, like glucose is. Here's glucose. So let's just have a little wee look at glucose here. Glucose. C, 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 and C. It's a 6-carbon compound, and it's formed up in a ring with an oxygen making that corner there. If you count up here, you can see 1, 2, 3, 4, 5, 6 carbons. 1, 2, 3, 4, 5, 6 oxygens, and 2, 4, 6, 8, 10, 12 hydrogens. C6, H12, O6. I think you've heard that formula before. Or we could then join 2 glucose molecules together, like this in maltose. And let's see how that's done. So we've got our glucose molecule here. By the way, these little dots here, these just represent carbons. So often in these sort of drawings, people don't bother to draw in all the carbons, but they are there. They're just on these corners here. And you'll see that each carbon, of course, has 4 bonds to it. 1, 2, 3, and 4. Each carbon having 4 bonds to it. So we've got 2 glucose molecules here. What can you do with these glucose molecules? You can take this OH from this carbon here on Mr. Glucose on the left. I shouldn't assign gender to glucose molecules. That's unfair. And we've got this glucose molecule on the right here. And we can take this H from it, and together we can take off our H2O and link our left and our right glucoses together. Here we go. We've got now an oxygen bridge between our 2 glucose molecules. We've got now what is called a disaccharide. A glucose molecule is a monosaccharide, i.e. it is the individual building block of carbohydrates. It is a carbohydrate itself, and larger carbohydrates will have glucose as their building block, as their monomer. You may learn that word from chemistry, monomer. And as we start to put our monosaccharides together, we form first of all a disaccharide, i.e. it's made out of 2 monosaccharides, and then we can start building more onto that. Now, this looks like quite a complicated structure to remember, and we wouldn't expect anyone to remember this at GCSE level. So have a look at this. This is a bit easier. I bet you can remember a hexagon. That's a hexagon, and we're going to take that to represent glucose. Oh, there's another hexagon. That can also represent glucose. So, what do? We react them together. We form a bond between them. Here is that bond. And we have our disaccharide plus water. This is our water molecule there. So we have our two glucoses coming together to form maltose. Oh, stop doing that, pen. That's maltose there. Just for our information, going this direction here is called a condensation reaction. It's called a condensation reaction because it's giving out water. If we want to go back this way, and we can go back this way, it's a very reversible reaction, we would call that a hydrolysis reaction. So hydrolysis goes back that way, and you need to add water, and that gives you back your two monosaccharides. The main function of our simple sugars is to supply energy for respiration. Glucose is really good for respiration, and we know the equation for respiration, don't we all? Well, if we don't, here it comes. It is C6H12O6 plus 6O2 goes to 6CO2 plus 6H2O. And that, of course, there is the formula for glucose. So they're really good fuel for respiration. Actually, they're not the only fuel for respiration, but they are a simple one to use. So we can then take our simple sugars, and here we've got our glucose monosaccharides, and we can join them together. This little symbol here just means it's a reversible reaction. And just as we join two glucoses together to make maltose, well, we can stick more glucoses together to make longer chains. And these longer chain carbohydrates are referred to as complex, as being complex. They are the complex carbohydrates. Now, they're still just made out of carbon, hydrogen, and oxygen. There's nothing actually that complex about them. They're just glucose monomers stuck together. That's all. And each time you make a bond, you give out a water. And each time you break it up again, you've got to take a water in. That's it, really. I'm just going to write this over here. So, joining them together, what did we say? We said that was a condensation reaction. And splitting them up again, that is a hydrolysis reaction. So with this you can make huge molecules. Let's start off thinking about starch. Now starch is the plant storage carbohydrate. And it's just, look, it's hexagons. It's hexagons joined together. It's glucose molecules just joined together. The glucose is the monosaccharide, and starch we would refer to as being a polysaccharide. A polysaccharide. Starch is large. It's really large. You can have them 500,000 glucose molecules long. That's huge. Because it's so large, it's basically insoluble, and therefore it doesn't dissolve and doesn't get lost from the cell. And also it means it doesn't affect the cell osmotically. That's really important, and if you haven't learned about osmosis yet, don't worry too much. You will learn about it in due course, but it won't make the water potential of a cell negative or more negative. It's also really compact, so you can store a lot of glucose in a small space, and it's not very reactive. You can keep it hanging around in your cell without worrying that it's going to go off and react with something or other. Whereas glucose, you shouldn't really leave that hanging around in the cell in its reactive form. So starch, that's really good, and that, let's just emphasize it, starch is for plants. Or rather, plants make starch. Of course, we can eat starch, and when we eat it, we then digest it down into twos. Hey, do you remember what we called two glucoses stuck together? That's right, that was maltose. So we digest it into maltose, and then we chop the maltoses up themselves into glucose. And that's what we do in digestion. But starch, made by plants.
Glycogen is really, really similar to starch. Now, these diagrams here aren't quite so, well, this one here certainly isn't quite so hexagonal, but you can see it's just glucoses again. It's just glucose. And we've got our chain of glucose running along here. Just the difference being that you can have branches coming off it. And glycogen is branched. It's the animal storage carbohydrate, and it's branched, which just means you've got lots of these ends here. And that's really useful for animals, because when we break down glycogen in order to release the glucose in it so that we can use it for respiration, we break it down from the ends. If you've only got one end, as you would do in starch, and we'll just think of the spiral form of starch for the sake of this PowerPoint, you've only got one end of that starch molecule, so you can only break it down quite slowly. If you've got 500,000 glucose molecules in it and you're breaking it down one at a time, it's going to take some time before you really get hold of all that glucose. But look at how many ends you've got here for glycogen, and that's really, really useful. It means you can break down your glucose quickly, and if you're an animal, you've got a high metabolic rate, so it's really useful to be able to get to your glucose quickly. Cellulose is the last carbohydrate we'll think about, and this also uses glucose. Now, there's a slight difference between the glucose used in glycogen, starch, with this one used in cellulose. For the sake of it, we use alpha glucose in starch and glycogen, whereas beta is used in cellulose, but hey, it's C6H12O6. It's the same jolly stuff, and they are easily convertible from one form to another. The point about cellulose is that it can be used to make plant cell walls. If it can be used to make plant cell walls, it must be pretty stable and pretty strong. And it is. You reorganize these glucose molecules slightly, and you find you suddenly have a very, very stable compound which won't dissolve and is really difficult for animals to digest. No animal yet has come up with an enzyme that will digest cellulose. So it's a fantastic compound to make your cell walls out of if you're a plant. And it's just glucose stuck together. They still are carbohydrates. And now let's think about proteins. Proteins are made up of many amino acids joined together. And I'm just going to draw one amino acid here. Here we go. Let's make sure I get it right first time. So far, what have we got? We've got a carboxylic acid group there. We've got a nitrogen over here. So that immediately means that, okay, what elements have we got in here? We've got carbon, oxygen, hydrogen. Well, that's just the same as carbohydrates. But we've also got this nitrogen over here. So, good to see you, nitrogen, and you have brought in something different into the mix. And then here, I'm going to write an R. What is that R group there? Well, that R counts as a variable group. That bit changes. And we have 20 amino acids that we use to make our proteins, and each one of those 20 has a different R group. Now, I'm going to draw another amino acid here, and we can see what might be about to happen, and maybe you'll be able to spot what's going on here, remembering what happens with our two glucose molecules when we put them next to each other. Okay, so these are our two amino acids. Now I'm going to call this one R1 and R2. So variable group 1, variable group 2. And now what we're going to do is we're going to join these two amino acids. Let's change our ink. Let's go to a lovely blue. And here we go. We're going to take these guys off here. They're going to become water. And what sort of reaction have we got going on here? We've got a reaction which releases water. Ah, it's a condensation reaction. And what are we going to get as a consequence? Well, here we go. We have joined our two amino acids together. Hopefully I've got enough room here. Here we go. R2. Okay, that's our second amino acid here, and then we'll finish our first amino acid here. Now, let's do a quick check. Has every carbon got 4 bonds to it? 1, 2, 3, 4, 1, 2, 3, 4. 1, 2, 3, 4, 1, 2, 3, 4. Good, every carbon's got 4 bonds. Has every nitrogen got 3? 1, 2, 3. 1, 2, 3. Yes, every nitrogen's got 3. Every oxygen's got 2. 1, 2, 1, 2, 1, 2. Good, that's good. Now this here represents 2 amino acids joined together. That is the start of a protein. And you can see we've got a sequence here, R1, R2. Now, we said there were 20 amino acids that could have been chosen from, and we chose these 2. But it could have been any one of those 20. Now, let's take a protein which is 10,000 amino acids long, which is a decent sized protein, yes. Decent size, but not the largest protein in the world ever, McEver. No. And at each one of those 10,000 places, you could have chosen 20 amino acids. Chosen one from one of 20 amino acids. That means there are 20 to the power 10,000 different possible proteins we could have made. We could have ordered those amino acids in 20 to the power 10,000 different ways. And when you're getting onto those sort of numbers, you're starting to say, well, if you used every atom that has ever been created in the universe, that has ever existed in the universe, and you use them to make protein and protein only, you wouldn't have enough atoms to make every possible protein 10,000 amino acids long. So you can see the sort of variety you can get with protein molecules. And let's have a look at them. Oh, before we do, we should just have a look at a simplified version of it. Here we go. This is our simplified version of proteins. These are amino acids. Notice they're different, but they've still got their means of bonding here, so these where the bonds can be made. And going this way, we're going to release water, and it's going to be a condensation reaction. And if we're going to break our protein back up into its amino acids, then that is going to require water. So water's got to go in, and that would be called a hydrolysis reaction. Hydro with water, lysis, splitting. Splitting with water, that's what hydrolysis is. Some details. All amino acids contain the elements carbon, hydrogen, oxygen, and nitrogen. Some also contain sulfur. Some amino acids also contain sulfur. 20 amino acids in all, giving an almost limitless number of possible proteins. What do we use them for? Okay, we use them for growth, repair, and making enzymes. Oh, making enzymes. Enzymes are proteins. They are lumps of protein. And we also find proteins in cell membranes, and we find them doing all sorts of things. Hemoglobin's a protein. We use that to transport oxygen. And the shape of proteins is extremely important to the way that they function. And in particular, we'll think about that when we discuss enzymes. The order of the amino acids determines the shape of the protein. That is why it's important that we see how many possible combinations of orders of amino acids there really are, because that means that there are a near infinite numbers of shapes of proteins as well, and that will determine what they do. So you end up with these wonderful three-dimensional shapes, 2 proteins. There we go, there's one. Here's another. This is a protein which would be used to cut up DNA. If you're really clever, you can see the DNA just in the middle of it there, and there's another bit over there. This is a restriction enzyme, and it cuts up DNA. It's a protein which cuts up DNA. And this is a structural protein. You can see it's much more regular in its shape, and that gives it a structural function, this being silk. Lipids. Fats and oils are lipids. Lipid is the category which includes fats and oils, also includes steroids and waxes. Lipids also contain carbon, hydrogen, and oxygen, just like carbohydrates, but much less oxygen than in carbohydrates. And they are made out of 2 types of molecule, glycerol and fatty acids, or rather the type of lipid we're thinking about is going to contain what contains glycerol and fatty acids. We have two broad categories of fatty acid, and that determines what type of lipid that we make. Saturated fatty acids are the ones that animals make, so in butter or in lard or just around the outside of a joint. I always think about food, really, when I think about lipids and biological molecules. They're solid at room temperature, and they're called fats, whereas those which are liquid at room temperature are called oils, and they contain unsaturated fatty acids. We'll have a look at that in just a moment. A large number of functions, and thanks for that. Large number of functions include, let's go back to their structure now, shall we? This here is a saturated fatty acid. You'll see every carbon has got as many hydrogens on it as it possibly can. This is a big alkane chain, whereas here we've got a double bond between these two carbons. That means that this is carrying two fewer hydrogens than it could otherwise, because the double bond is taking up bonds which could be used to hold hydrogens. Therefore, this is not saturated with hydrogens. It's an unsaturated fatty acid. And that's an alkene. This molecule here is an alkene, or this part of it is an alkene. Here, we've got two areas where there's a double bond, and so this is a double unsaturated fatty acid. Back to their functions. Large numbers of them, and they include a store of energy. They are fabulous for this. Absolutely fabulous. So they store much more energy per gram than carbohydrates or proteins. And so, when our grizzly bear is getting ready to hibernate, they will be at their largest body mass because they have stored up so much lipids. So they'll gorge themselves in the summer, and when the salmon start leaping up the springs and the streams, they'll gorge themselves, pile on lots of fat, lots of wonderful lipid, so they can store that energy for their hibernation over the winter. Of course, hibernation is over the winter. That's the derivation of the word. Then, next function, insulation. Very important for chaps and chapesses like these walruses here. These walruses, you can see, are extremely well insulated. They are lying on an extremely cold, rocky beach at the moment. But you can see they're pink. They're pink because they're vasodilating. They're overheating because they are so well insulated. They're really well insulated because they are normally found swimming in icy waters. We also need them as protection from impact. We have some over our kidneys to protect our kidneys from impact. And they are a very important part of the cell membrane. The cell membrane is mainly lipid. This is the lipid diagram for your notes. And here we have glycerol. It looks like an E. Yeah, okay, fair enough. But what it has is it has 1, 2, 3 regions where it can bind to fatty acids. These fatty acids are just largely alkane chains with a carboxylic acid group at the end of it here, so that they can bind on to the glycerol molecule. So 3 times fatty acids and 1 glycerol. Now you can join them together. So we're joining together going up the page this way in, guess what? In a condensation reaction. That's right, people. We get another condensation reaction. We get water given off by it, and if we want to split our lipid back into glycerol and 3 fatty acids, we had better put my arrow going this way. Better put water into it, and that will be a hydrolysis reaction. This hydrolysis will be catalyzed by the enzyme lipase, and we'll learn about that in the digestion module. DNA, nucleic acids, including DNA, is the last big category of biological molecule, and we're going to look at DNA properly in another topic. But it's a wonderful molecule. Contains carbon, hydrogen, oxygen as before, but also nitrogen and phosphorus. Tests for biological molecules. This is Benedict's solution. possibly plus some little sugars in here, in some glucose in there. And this is what happens if you heat Benedict's solution when there is glucose present in it. It becomes brick red. So the color change for a positive Benedict, where you'd heat it at 90 degrees C for 2 minutes, would be a color change from blue to brick red, and that would tell you there was glucose present in that solution. Now I've written all over this text, but you can figure it out. Here's a standard procedure for you. But yeah, you whack in Benedict's solution, or add, which is the common term. You heat it using a water bath to 90 degrees C, and then you'll see a color change from blue to brick red. Although it could just change to a green or an orange, it can change to those other colors as well. This is the iodine test for starch. It's even simpler than the Benedict solution. You get your test solution, you squirt some iodine in. Iodine is this kind of yellow color here. And if there is starch present, we have this color change from this orangey yellow of iodine to a blue-black color. In fact, you can just say black. If you're worried that you might get confused with the blue looking like Benedict's, you can just say black. Here we go. I've written all over this as well, but not quite so bad. Not bad at all. So brown to blue-black. There we go. So, tutorial questions. Have a read of these tutorial questions, pause the video, and if you need to, go back through the video to try to find the answers. I'm going to sign off now. Hope you found the video helpful. Thank you.